All right, good evening, everyone. This is the fourth week we are looking at the R coding practices and ideas. And this week, I would like to introduce you a couple of ideas about annotating gene groups. And again, I won't go through the whole details and all the analysis, all the methods, but I would like to just highlight a couple of points. So just again, when I was at the university, which was quite a long time ago, we still had this kind of old school research that you were working with a maximum of handful of genes, or you just tried to interpret the effect of a single gene or gene product. And you just mutated or did some alterations and did old school mutagenesis, and you have seen the results immediately. But nowadays, so the main point here is that basically there was like a direct connection, one gene connected to one phenotype. And this is coming from the traditional view how genetic works, that the original idea during the first half and the middle of the 20th century, that each of the phenotype, what we can observe in an organism, has a corresponding coding gene, which is obviously impossible in the modern context of genomics, since we know that in, for example, in human genome, we have roughly 20,000 genes, a little bit more, but of course we can find more than 20,000 phenotypes in a human being if you put together behavior and physical features and whatnot. So, but regardless, that was the idea, that every phenotype has a corresponding gene which is defining that phenotype. And you can still hear or you can still recognize this idea, this approach, when you read this kind of popular news articles that they have found the gene for longevity, or gene for obesity, or gene for Alzheimer, or gene for cancer susceptibility, which is obviously not working like that. We have now an understanding that the connection between genotype and phenotype can be very, very complex. And exactly this is the challenge what we encounter in modern molecular biology research, that very often when we try to investigate a phenomenon, we recognize that there might be a whole panel or a list of genes which is somehow involved. So when you study some kind of, for example, some kind of disease, you obviously try to find that, okay, what are the genetic, if it's proven that disease is genetically defined to a certain extent, then you gather samples from a healthy group and a disease affected patient group, and you try to find some kind of connection. And very often the case is not that, yes, there is this single gene, if it's mutated, then you have the disease. Admittedly, there are such cases, but those are more the exceptions. So very often the outcome is that, hey, we have a big panel of genes, and which are significantly, has a significant association to some kind of phenotype, for example, a disease state. Then we should try to say something about those genes, because when it is a single gene, then okay, it's a transcription factor. If it's mutated, then it's not doing its job. Genes are not turning on and off. It's relatively straightforward to find some kind of biological explanation for the mechanism. But when we perform an experiment, compare disease patient group and healthy control group, and we find that, yes, there are 150 genes affected or with a different genotype, then we are scratching our head that how to interpret these kind of gene lists. So there are a couple of tools what we can do, what we can use in this kind of situations. And basically, enrichment analysis is one of the more established ways to basically interpret gene lists and saying something meaningful, biologically meaningful about a group of genes, which are very often coming from these kind of high throughput experiments. It's maybe you might be or you might be not familiar with the term enrichment analysis. It is because it is a collection of methods, and very often it is applied to a certain kind of annotation which can be associated with genes. And the more known name for these methods are coming from those annotations. So enrichment analysis in theory can be performed to any kind of gene annotation, but most often it is performed for example, for gene ontologies. Then it's called ontology analysis. When it's applied to pathways where the gene products are taking part, then it's called pathway analysis. When it's applied for transcription factor or microRNA binding sites, then it's called transcription factor analysis or microRNA binding analysis. So basically, all these kind of methods, they are applying the same kind of statistical approach. And the basic fundamental question these methods try to answer is what this is distinguishing this particular group of genes compared either to a reference group of genes or the entire genome. So, for example, if we perform this for gene ontologies, as probably the most often the case, we are asking that, okay, I have done some kind of high throughput transcriptome analysis, for example, mRNA-seq, and I've got a panel of 100 genes which are differentially expressed between the groups I'm investigating, for example, patients, disease-affected patients and healthy control patients, and which are the gene ontologies which are significantly associated to my target genes. Because if I find a couple of such ontology terms, then I can tell that, for example, if those are kinases, then probably the kinase phosphorylation mechanism is affected somehow. Or if they are, for example, associated with adaptive immunology gene ontology term, then we know that the adaptive immunity is affected in those patients, and so on and so on. Same way, if we are performing enrichment analysis using pathways, for example, CAG pathways, if you find that that panel of genes, what you have got from your experiment, it is significantly associated to one particular signaling pathway, then of course you have a very good biological explanation that what is happening in your experiment, what is distinguishing your different groups. Okay, So enrichment analysis is a collection term for this kind of association studies. Basically, there are two different methods, and I would like to highlight similarities and differences between those two methods. One is called overrepresentation analysis, and the other is called gene set enrichment analysis. Both are subcategories of this larger enrichment analysis method. They are working differently when you apply the algorithms, but basically they are serving similar purposes. Both of the methodologies they can be applied to any of these gene annotations, and not even restricted to those examples, any kind of annotations which is meaningful in your context. For example, once I was involved in a research project where we have identified a list of genes which went through positive selection during the evolution of mammals compared to the rodents, and we used an ontology type called disease ontology, and we tried to figure out if there are some diseases which are especially prevalent in mammals because of this accelerated evolution, that kind of things. So disease ontology is not equal to gene ontologies. It's a own kind of ontology used in health research. We just tested if the idea worked there or not. So just again, otherwise there are like a million kinds of ontologies developed during the past couple of decades, which are in use in molecular biology. So be aware that gene ontologies are just one of those. Admittedly, they used most often, but no way that that would be the only example of ontologies. So ontology just means restricted or defined vocabulary. You can use it in any domain, and they are usable in enrichment analysis, given that they are describing genes. All right, I have drifted a little bit too far away from the main topic, which is enrichment analysis. So overrepresentation analysis and gene set enrichment analysis. Why we do have two different methods? Because they have a slightly different focus and slightly different requirements. So overrepresentation analysis is working with two gene lists. Basically, you have a target gene list, for example, which is coming out from your RNA-seq experiment list of genes which show significantly different expression patterns between your groups. And there is a background gene list. The background gene list is a wider list of genes. Very often it is the entire genome, so all genes identified in a certain genome. But not always. Like again, in one of my research projects a couple of years ago, it was shown that we were studying a group of 1,000 genes in different genomes, which were being labeled as the immunome. So those genes which were involved with different kind of immune processes, their products. So we used this immunome 1000 genes as a base set, and in different experiments we identified subgroups, and we performed this kind of enrichment overrepresentation analysis, where the target group was target gene list was a subset of this immunome. So in this particular example, the background gene list is not the entire genome, but nowadays it's just so that very often you take the entire genome as the background list. All right. So from statistical point of view, you perform a hypergeometric test. It is a statistical test, very similar to what we have practiced in the statistics course. So it's the now hypothesis is that the distribution of the terms is the same in the background set and the target set, and you can calculate the significance level or the p-value, and that is the traditional threshold of 5%, and under that you can say that one particular ontology term is distributed differently in the background set and the target set. In the Moodle video, among the Moodle videos, there is a quite detailed explanation how this works. Basically, the textbook example, if you have a target gene list consisting of, for example, 50 genes, and you have a gene ontology which is in the entire genome have 60 representation, and 40 of those are on your target gene list, that it is probably not an accident. Probably your experiment has something to do with that particular ontology, because it is significantly enriched in your target gene list. So it's important to understand that when you perform enrichment analysis, you do a statistical test for practically all the ontology terms what you find in meaningful in a set, and remember what we need to do when we repeat the same kind of statistical tests again and again and again. Of course, we have to adjust p-values, otherwise we will find enriched terms just by chance, because we are doing so many tests. What is coming out from overlap representation analysis is very often a ranked list of ontology terms, where the top terms are those which are most heavily associated with your target gene list, and you are ordering them according to this strength of association. Okay, so overrepresentation analysis, target gene list, background gene list, and hypergeometric test. In reality, you do not even need anything specific to implement this in R, because hypergeometric test is a generic hypothesis test. So you can do it with basic R without installing any packages. But of course, there are many procedures or many experimental method specific R packages which are implementing this kind of wrapper functions, which makes it very convenient to implement this. The other method, what is under this big term enrichment analysis, is that the so-called gene set enrichment analysis. Here you have only a ranked gene list. So a ranked gene list is very often coming out from differential gene expression analysis, which is saying that on the top of this list you have genes which are most heavily associated with one of the groups what you are comparing in your experiment, second, third, fourth, and so on. So there is a natural rank of the lists, usually based on some kind of association statistics. And this ranked list can be investigated, that what is the probability that the order of that gene of that list is modifying the random distribution of ontology terms along the ranking of that gene list. Of course, if the ranking of the gene list and an ontology term are not associated, then these terms are, or genes annotated with that particular term, they are appearing randomly on your ranks of ranking of genes. But if this ranking in some way affected by that ontology term, then you will see that those genes associated to those terms are grouped either the top or the bottom of your rank gene list. So to mention some kind of example, so if the ranked gene list is coming as usual from some kind of differential gene expression study, then on the top of this ranked gene list you will have those genes which are overexpressed because of the experiment that you have performed, and at the bottom of the list, ranked gene list, you will have which are repressed because of your experiment. For example, if you are investigating the effects of some kind of drug compound, on the top of the list you will see which genes which will be most probably upregulated when you apply that drug compound, and on the bottom of your list you will have those genes which are downregulated when you apply that kind of drug compound. And if there is some kind of ontology term which is associated to those genes which are upregulated, they will appear mostly. Genes which are associated to that ontology term will appear mostly on the top of your gene list, and so on. So if that term what we are investigating, for example, a pathway, then you can give this kind of statements that yes, when I apply this drug compound, this particular pathway is mostly upregulated, so starting to work better or at higher efficiency. And if a pathway term is associated to the downregulated genes, then you can say that yes, we see the effect because this particular pathway is downregulated or pushed down by this drug compound. So that's how this kind of analysis can help you to interpret pure gene lists, because this kind of statements cannot be made based on just looking at the gene lists which are coming out from your experiments. And this is the reason that this kind of enrichment analysis is most often applied at the interpretation phase of a project or an analysis. So most of research projects has their standard internal logic. So first, most often you do some kind of exploratory analysis, and then you produce this kind of huge gene lists. Then you try to interpret them, and this is one method, one way how you can interpret those gene lists. And of course, then the story goes forward. So if you have seen that, for example, a drug compound is working because it is pushing down some kind of pathway, then you plan some kind of targeted experiments which can prove this kind of hypothesis. Okay, so this is the place when such enrichment analysis can help you with your research. Yeah, I think in the next couple of slides there are examples of different kind of annotations which can be used in enrichment analysis. One of those is gene ontology. I don't particularly want to go into the details of gene ontologies because I know that on one of Matti's, Dr. Tolvanen's course, there was a very detailed material about gene ontologies. You remember that there are basically three kind of ontologies under the umbrella term of gene ontologies, describing cellular components where gene products are taking part, molecular functions, and biological processes. So I don't want to go into those details. I would like to just mention that gene ontologies themselves are not independent from each other. They have relationships with each other. So one particular ontology can be a part of a larger context ontology, or it can be a super group of ontologies. So these ontologies are interconnected with each other, and this all of them, if we draw such kind of diagram, how they are connected to each other, what are the hierarchy, what is part of, and what is an ISA connection, as you can see here, then basically we can build up this kind of graphs of gene ontologies. And this particular kind of graph is called the directed acyclic graph, or DAG, because it looks like this for a small set of ontologies. So each rectangles are representing a gene ontology, a biological process, and molecular functions examples. And you can see that they can be like related to each other. So a response to stress is a subcategory or sub-ontology of a response to stimulus, and response to stress is a super group or higher in hierarchy than a response to wounding. So it's important, or I find it important, that you realize that this is the ontology graph itself has some kind of specific structure, and this can be used in enrichment analysis. So on the actual slide, what you see is results from such kind of enrichment analysis. The red ontologies, they are associated significantly to genes on a target list, or this is an outcome or a result from overrepresentation analysis kind of method. You can see that, for example, immune system process is associated to 26 genes on that target list, and you can see the adjusted p-value. So you can see that this structure, this directed acyclic graph, has very interesting consequences for the analysis. So very often, if target gene list shows a heavy association to a term at higher levels, for example, immune system processes, then very often many of the subcategories, sub-ontologies are also significant. Like immune system processes is a high-level ontology. It's closest to the root biological processes. So immune response, regulation of immune response, positive regulation of immune response, these are subcategories. You can see that all of these are red, so they are significantly associated with the target gene list, what were analyzed in this experiment, or what was the output of that experiment. So we can say that probably those genes which were affected by the experiment are part of immunoregulation. But you can see that it's not always the case. There are certain terms which are black, so not showing significant association, even though intuitively they should, like the positive regulation of immune system process. It's not significant. So it is good if you perform overrepresentation analysis, you find the associated categories and you try to go as low in the hierarchy, so to find the most specific terms which are significantly associated to your experiment. Because if you say that, okay, I performed an experiment, I got a panel of 70, 80 genes, I performed overrepresentation analysis, and the outcome was that response to stimulus or immune system process ontology terms are enriched in your target gene list, and maybe it's not very meaningful, or it's too general to have some kind of conclusion for your experiments. But if you say that inflammatory response 14 genes, then yes, it can be indeed. So you can know that whatever was your actual experiment or study, it is heavily affecting inflammatory response, which is now an opportunity to move forward with your research project, make advanced or targeted experiments. Okay, so this DAG, directed acyclic graph structure of ontologies, offers this as an extra opportunity because the ontologies themselves have relationship with each other. Yeah. So in the Moodle video material and the written material as well, you will have a detailed example-based explanation how to do the actual calculations for the overrepresentation analysis, and how to calculate the hypergeometric test. And just again, in basic R, without installing any further packages, you can perform the hypergeometric test using the p-hyper function. It is actually quite didactical to do it at least once, try out how it looks like. So just again, if you calculate the enrichment of a single ontology term in your experiment, then you get a raw p-value, which is good. But if you perform it as it usual, so you perform the overrepresentation analysis for all ontologies in the ontology database, then you will perform probably a couple of thousand of hypergeometric tests. So you will need to adjust the p-values as discussed on the statistics course. Okay. Yes, multiple hypothesis testing. This is just a side note. We covered this in the statistics course, that in molecular biology, basically we use only two methods for correcting for the raw p-values when we perform multiple hypothesis testing. Either we use the Bonferroni method when we have to be very sure that what we identify significant, it is really significant. When we can be more relaxed, then we use the Benjamini-Hochberg method, or also known as the fast discovery rate. Of course, when we perform enrichment analysis, it's very, very rare that we have to be very strict and we have to use the Bonferroni method. it is reserved more for cases when you analyze the effect of drugs, and really patient lives depend on the outcome of your calculations. So in research, and especially in enrichment analysis, it's quite okay to use the Benjamin-Hochberg method, which is much more relaxed. Yep. In basic R, again, the p-adjust function can be used to correct for multiple hypothesis testing. Basically, it is a very interesting function because it accepts a vector of p-values. So, for example, if you calculate, put your data into some kind of data frame and calculate the p-values for all the ontology terms what you want to analyze, you will have, for example, 2,500 p-values. You just assemble that as a vector, as a numeric vector, and just feed it to the p-adjust function, and that's it. You will get back a vector of adjusted p-values, and it is very, very quick, and it doesn't need any further information. Also, the good news is that the default, the p-adjust function has a method parameter, which where you have to tell that which method do you want to use. The options are, for example, Bonferroni or Benjamin-Hochberg or FDR. These are the ones I mentioned previously. So that's how it works. But if you do not provide a method, then it uses, if I'm correct, but I will just double check, which is the default. Yeah. The FDR is the, I think, the FDR. So the force discovery method, alias Benjamin-Hochberg, is the default method. So if you do not provide anything, that's what you get. Gene set enrichment analysis. So the other methodology, the other algorithm, what you can apply here. Just again, you need as an input a ranked gene list, and basically it's a nice graphical way to do. So here on the left-hand side, this is an illustration. We have a ranked list of gene IDs. So the numbers on the left, they are representing gene IDs from the NCBI database, and it is marked that which of those are associated with the MAP kinase pathway, which is a KEGG pathway. You see that some of them are, and this is just the top of the list of 2,500 genes which are associated with, which were analyzed in this exercise, in this gene set enrichment analysis exercise. So basically, this is a huge table with 2,500 rows, and there are roughly 300 genes which are associated with the MAP kinase pathway, and some of them yes, some of them no. So what we do is, actually, I find it visually very easy to interpret. So we have the ranked list, so first, second, and so on, and we turn this list to the side, and that will be our x-axis. So this is the first gene, then the second, third, and so on and so on. And what we do is basically we calculate some kind of score, which is called the running enrichment score, which is working so that on the top of the list, the value of the running enrichment score is 0. And we are stepping one by one down on the list, and we found or encountered a gene which is associated with the MAP kinase pathway. Then our running enrichment score is stepping up, so we are adding some kind of value. And when we encounter a gene which is not associated with that particular ontology term, then we are stepping down. So we are subtracting a value. So we plot the running enrichment score as a function of the ranks of the genes. And when in the regions of this list which are associated mostly with MAP kinase pathway, the score is going upwards, and at parts where it is not, then it is decreasing down. So this is just like a artificially assembled gene list in this example. So you can see that the top of the list, on the top of the list, so on the left-hand side of the x-axis, this running enrichment score is mostly climbing, and we can say that most of the MAP kinase pathway genes are associated with the top of the list. So we can just take a peek at the shape of this diagram, and we can tell that which part of the list is associated with those terms, if at all. So if the list is not associated in any meaningful way with that particular ontology term, then obviously these genes will be randomly dispersed along the x-axis. So this curve is just going up and down around 0. So there is no strong peak or a deep valley. If it's associated very heavily with the top of the list, then there will be a big peak. And if it's associated heavily with the low end, so the bottom of the list, then there will be a very deep valley. So there are this kind of curves what you might see. So I use just again an artificially assembled list, but the analysis is real. So you can see that in case of MAP kinase signaling pathway, what you have just seen, there is a peak, and in case of Huntington disease, there is a valley, and so on. And oxidative phosphorylation, there is a valley and a very deep bottom. So we can say that this particular term is associated with the top, and this term is associated with the bottom of that gene list. So that's why it's important that the input gene list here is ranked, while in overrepresentation analysis is just a bag of genes. It doesn't matter that if there was some kind of order there or not. Yeah, I always, back to gene set enrichment analysis. So what you see here is an analysis of a single ontology term. This was a KEC pathway analysis, so a MAP kinase signaling pathway. But in practice, when you do this for real, you analyze for all the annotation terms in the set what you use. So, for example, all the pathway terms in KEG, or all the ontology terms in gene ontology database. And again, you do these calculations for hundreds or thousands of times. So, and the analysis itself is providing actually a p-value. So you have to adjust it again because of this multiple hypothesis testing. Although, strictly speaking, this is not a hypothesis test, but still the p-value has to be adjusted the same way. So at the end, you get a group of ontologies which are associated to one or other part of your gene list. What I would like to stress as a last idea in this demo or in this session that the last step in an enrichment analysis is to try to interpret and try to understand the significance of the terms which were coming out from your analysis to your experiment. So there is a very important distinction between a statistically significantly enriched annotation term and a biologically relevant one. So there are, so for example, if you are studying, if you study, for example, gout, which is a disease, and it is known to have a defect of purine metabolism. So, of course, if you collect samples from gout patients and healthy control, and you do a differential gene expression analysis, you will get a list of genes, and you perform enrichment analysis, you won't be surprised that purine metabolism will be probably among the heaviest associated with your gene list. So this purine metabolism ontology term enriched in gout patients is a statistically significant enrichment, but its biological relevance is practically zero because it is a known phenomenon that gout is a defect coming from a defect of purine metabolism. So you have to sort of try to interpret it. Is this result meaningful for my experiment? You just cannot mechanistically publish and say that, yes, this is the reason, and that's it. Okay, some further explanations in the Moodle videos. I will stop here. So I will ask if you have any questions to this material.
No, don't have any.
Can you repeat it a bit louder?
No, I don't have any.
Okay, no questions. All right, then that was what I wanted to especially stress this week. So I will stop the recording.